Hello, and welcome to today's Chapter 6 video. We're going to talk about viruses and prions in this chapter. And so these are a couple of the giant microbes that we find representing the viruses and prions. This, of course, is our favorite little guy right here, the coronavirus, and this is representing bovine spongiform encephalopathy, one of the major prion diseases that we see. Viruses were very hard to actually discover. In fact, it wasn't even until the very late 1800s that viruses were even discovered. Pasteur actually developed a vaccine for rabies, but they really didn't understand what viruses were even at that time. Pasteur actually coined the term virus, which was Latin for poison. A couple of Russian individuals, and I can't pronounce their name, but here they are, actually discovered a plant virus, the tobacco mosaic virus. The tobacco disease actually was causing major economic losses in tobacco plants, and so of course they wanted to figure out what this was. Leffer and Frosch actually discovered then foot and mouth disease in cattle that is caused by a virus. What they actually found was that there was something that was filterable, that even if they put something through a filter that was small enough to take out all of the bacteria, in the filtrate there was still this infective particle. And so infectious fluids were passed through filters designed to trap the bacteria, but yet they could still cause infection in the plants. This cell-free filtered fluid, though, remained infectious, and so they actually came up with this term that was for the virus. This proved that an agent smaller than bacteria was the actual cause of this plant particular disease. So true or false, viruses can be cultured using the same methods as bacteria and fungi. Pasteur was the one who developed a vaccine for which virus? And viruses were actually discovered by filtering infectious fluid through a filter designed to trap what? Now, viruses can infect any type of cell. They can infect bacteria, algae, fungi, protozoa, plants, and animals. Are they truly organisms and are they alive? Well, this is actually a very debatable question because we're not really sure. What role did viruses play in the evolution of life? What are their distinctive characteristics? How can a particle so small and so simple on a genetic standpoint and seemingly so insignificant be capable of causing disease and death? And what is the connection between viruses and cancer? What role did viruses play in the development of all other? So, first of all, some unique properties about viruses. They are infectious particles, but they are not truly organisms. I will refer to viruses as active or inactive rather than alive or dead because truly viruses are not living organisms. They simply don't have all of the characteristics of something that is living. We do know that they are obligate intracellular parasites because the one thing that they do share in common with living organisms, the ability to reproduce, they must do inside a specific living host cell. They must instruct the genetic and metabolic machinery of the host cells to actually make and release new viruses. And so here's some property of viruses. They are not cells. They do not have the properties of cells. They are obligate intracellular parasites of all other living organisms. They do not independently fulfill the characteristics of life. They are inactive macromolecules outside the host cell and active only in the host cell in reproduction of the virus. They have a basic structure of a protein shell known as the capsid that surrounds a nucleic acid core. They are ubiquitous in nature and have had a major impact on the development of all biological life. They are ultramicroscopic in size, ranging anywhere from about 20 nanometers to about 400 nanometers in diameter. They carry either DNA or RNA, but never both. They can also have double-stranded DNA, single-stranded DNA, single-stranded RNA, or double-stranded RNA. They carry molecules on the surface of the capsid or envelope that determine specificity for attachment to a host cell. Most viruses are actually very host cell specific. They multiply by taking control of the host cell's genetic material and regulating the synthesis and assembly of new viruses. They literally hijack their host cell's protein synthesis in order to make more viral particles. They lack enzymes for most metabolic processes, and they lack machinery for synthesizing their own proteins. And this is why they have to have a host living cell. So true or false? Viruses are alive. Well, while they do have some characteristics such as reproduction of a living organism, they truly are not alive by the true definition of something being alive. What is the predominant view among scientists as to the nature of viruses, and how much do we really know about viruses even today? What percentage of the bacterial chromosome is actually composed of viral genes? And list and describe 5 unique properties of all viruses. So again, these are ultra microscopic in size. They're smaller than the average bacterium in general, and electron microscopes are required to detect them. For example, parvovirus is about 20 nanometers or 0.02 micrometers in diameter. Coronaviruses are also in this range. The mimiviruses are about 400 nanometers in length, and these are actually larger than some of the smaller bacteria. Cylindrical viruses can be somewhere between 800 to 950 nanometers long, but only about 15 nanometers in diameter. And so this is just simply a size comparison. So here's a budding yeast cell that's about 7 microns in diameter. E. coli is about 2 to 3 microns long and about 1 micron in diameter. Streptococcal cocci are about 1 micron in diameter. And the rickettsial cells, which are bacterial cells, actually are about 0.3 microns. The mimivirus is about 450 nanometers, and then you can actually go down through here and actually find that poliovirus, about 30 nanometers. Yellow fever virus is about 22 nanometers. And that's not much larger than the hemoglobin molecule itself found in our red blood cells at 15 nanometers. So here's some pictures of the viruses. And this is simply an adenovirus. Here's another virus. This is one of my favorite viruses, even though it's a very deadly virus. It is the only one that have this kind of bullet shape, and that is rabies virus. And this is actually one of the longest cylindrical viruses that we see at about 950 nanometers long, and this is Ebola virus. Viruses bear no resemblance whatsoever to cells. They completely lack any protein synthesizing machinery such as the ribosome. That's why they need to borrow from their host cell. They need only those parts required to invade and control a host cell. So they have an external coating, a core containing either DNA or RNA. And this is just simply the drawing of some of these viruses. So we have what we call a naked nucleocapsid virus that simply has the protein capsid coat surrounding a nucleic acid core. Or some viruses are what we call enveloped. And the envelope is actually a phospholipid membrane that's probably picked up from the host cell, and typically the envelope will have spikes, and these spikes are going to be important in being able to attach to a host cell. So the capsid is found surrounding all virons or virus particles, and this is made up of single proteins called capsomeres. The envelope is not found in all viruses. The central core, again, is either DNA or RNA, never both. And in some viruses, they do carry enzymes that are necessary for their reproduction once they get into their host cells. But this depends upon what type of virus it is, whether or not they're carrying any enzymes, and what those enzymes actually are. The capsid, as I said, is this protein shell that surrounds the nucleic acid core. The nucleocapsid is referred to as the capsid and nucleic acid together. The envelope, if the virus has one, is usually a modified piece of the host cell's membrane. This is often picked up as the virus buds outward from the host cell. A naked or non-enveloped virus consists only of a nucleocapsid. The spikes are found on both naked and enveloped viruses. These project from either the capsid itself or from the envelope. These allows viruses to actually dock with their host cells, and this is also what usually makes viruses very host cell specific. A viron is a fully formed virus that's able to establish an infection in a host. So once the virus is assembled within the host cell and released, we then refer to it as a viron. So here's actually a non-enveloped virus, and these spikes are simply coming off of the capsid itself. Or again, that envelope virus where we find those spikes on the envelope. The capsid is made up of these capsomeres, and these are identical protein subunits that spontaneously self-assemble to form the capsid around the nucleic acid core. Sometimes they take on a helical shape, and these are oftentimes rod-shaped capsomeres that form a continual helix around the nucleic acid, or icosahedral capsid, which are three-dimensional and usually 20-sided figures with 12 evenly spaced corners. And this is one way that we can help identify what type of virus it is. Do they have a helical or an isohedral capsule? So this is just showing, again, one of the helical capsules that's forming around the nucleic acid core. A naked virus of the helical shape is shown here. And this is actually a helical shape surrounded by a capsid, which then has an envelope around it. Just a little better, closer view of these particular viruses. The isohedral capsules actually make up then these triangular shaped faces on the surface of the capsid. And you can certainly kind of identify these triangular shaped structures right here on the surface of this virus. Just showing some more pictures of these isohedral viruses. And again, this actually shows then these triangular shaped faces on this naked virus. There can be complex capsid, and these are found primarily in a group of viruses known as the bacteriophage or bacteriophage. These bacteriophages actually infect bacteria, hence the name. And they can have multiple types of proteins within the capsid. They take shapes that are not necessarily symmetrical. And this actually shows one of these really bizarre shaped uh, bacteriophages. This is the nucleophage right here. So showing the capsule with the nucleic acid. They then have a collar and a sheath. They have tail pins and a base plate, and then they typically will have 6 of these tail fibers. So they kind of look like a weird shaped mutant spider shape. The viral envelope, if again the virus has it, is usually composed of the membrane of the host cell. And this can either have come from the cell membrane itself or could be actually part of the nuclear membrane of the host cell. Regular membrane proteins are replaced, however, with viral proteins. And the spikes, if they have them, protrude, and these are usually glycoproteins essential for attachment to the host cell. So just showing a naked or non-enveloped virus here with the capsid. This would be the capsid in the red, and then the envelope is around the outside. So another way that we can identify viruses, are they non-enveloped or naked or enveloped viruses? The genome of the virus is the full complement of DNA or RNA that's carried by the virus. And again, viruses contain either DNA or RNA, but never both. The number of viral genes is very small in comparison to even that of a bacterial cell. They possess only the genes necessary to invade the host cell and redirect the activity of the host cell in order to make more viruses. Now, we can see that again, they can be single-stranded or double-stranded DNA or RNA. If it's single-stranded RNA, we can have positive sense RNA carrying viruses, and the single-stranded RNA genomes carried by these viruses are actually ready for immediate translation into proteins once the viral genome is inserted into the host cell. Negative sense RNA, however, these RNA genomes need to be converted into the proper form, basically copied to the opposite strand, which would become the positive sense RNA, in order to be made into proteins once these negative sense RNAs are injected into the host cell. So here we can see some differences. Double-stranded DNA carrying viruses, and a good example of this is the variole virus, or the virus that causes smallpox, and herpes simplex 2, which causes genital herpes. An example of a single-stranded DNA carrying virus is the parvovirus, and in humans this particular virus causes a skin condition called erythema infectiosum. This is not the same parvovirus, however, that causes an intestinal disease in animals. Of the RNA viruses, the single-stranded positive RNA-sensed viruses, a good example here would be poliovirus. Single-stranded negative RNA-carrying viruses, a good example is influenza. A double-stranded RNA-carrying virus is actually rotavirus, and rotavirus is an important virus in that it causes a gastroenteritis in most patients, and this is the #1 cause of gastric diarrhea around the world. And then the single-stranded RNA reverse transcriptase carrying viruses, which we'll talk about reverse transcriptase here in a moment. An example here is the human immunodeficiency virus that causes AIDS. So how do these viruses actually replicate once they get inside their host cell? Well, it depends on what type of genetic material you are carrying. If you are a double-stranded RNA carrying virus, which is of a class 3, and we'll talk about those classes later on, this means that you have both a positive and negative sense of RNA. So the transcription will occur from the minus strand. And so you actually will see a positive strand of messenger RNA that will be made, and this will then be used for viral protein synthesis. If you were a positive stranded single RNA carrying virus, which is a class 4, this is already in the correct form, and it simply is used as the messenger RNA strand to translate into viral proteins. Single-stranded negative sense RNA carrying viruses, which are class 5, we simply will make a copy of the negative strand of RNA to a positive strand that then is used for translation. If you're a single-stranded RNA positive sense carrying strand, that's a retrovirus, which is in class 6, then they carry a reverse transcriptase enzyme that will convert that single-stranded RNA into a complementary double-stranded DNA, whereas that then will be transcribed into, again, a messenger RNA that will be used for translation into viral proteins. Now, some viruses do carry their own enzymes. For example, there are viral polymerases that will be used to synthesize DNA and RNA in the host cell. There are also replicases that can copy RNA, such as the negative sense single-stranded RNAs that then use this replicase to create the positive sense strand for messenger RNA in the host cell. And then there are those reverse transcriptase carrying viruses. And the reverse transcriptase will simply synthesize DNA from the RNA. Now, some substances are picked up actually from the host cell. Adenoviruses, oftentimes as they are being assembled, will pack along some host ribosomes that they will then carry to the next host cell. They cannot, however, use the ribosomes on their own. And retroviruses borrow the host's transfer RNA molecules, again to carry to the next host cell. So, true or false, viruses can be seen with the light microscope. Viral capsids are constructed from what subunits? And can you describe the structure of the isohedral, helical, and complex viruses? And list 3 unique forms, and that should say of viral nucleic acids. Now, how do we classify viruses? Well, there's actually a very informal classification system that's still used today. And this is, are you an animal, plant, or bacterial virus? Plant viruses do not infect animals or bacteria, nor do bacterially infecting viruses infect animals or plants. Are you a DNA carrying virus or an RNA carrying virus? And is your capsule helical or isohedrical? Now, the formal classification system, known as the Baltimore classification system, this looks at structure of the nucleocapsid, the chemical composition, and primarily, again, DNA, RNA, positive sense, single or negative sense, double-stranded, single-stranded, and what are the similarities in genetic makeups within the viral family. We typically classify viruses according to their family, and the suffix viridae refers to virus family. So, such as, for example, Poxviridae is a family of viruses. If we're looking at, quote, the genera or the genus of virus, we simply use the suffix virus. So, herpes virus is the genus, while herpes viridae is the family. Now, there is a group of nucleocytoplasmic large DNA carrying viruses that are all closely related to each other, but actually distantly related to other viruses. And this may actually constitute a new order of viruses. And we're not going to really spend a lot of time with these nucleocytoplasmic viruses simply because we don't know a lot about them even at this point in time. So let's look at some classification of viruses. And again, I want to focus more on the family name than anything else. But for example, within the family Herpes viridae, we have the herpes viruses primarily. And we'll go through some of these a little bit later on, but I'm just going to point out a few here. So in the Rudiviridae family is the rabies virus. Paramyxoviridae family, we have measles. The Fileoviridae family is Ebola, among others. The Togaviridae family, the rubella virus, which is the other form of measles that we see. The Poxviridae family, and this is a group of, and these all are DNA-carrying viruses. Poxviridae family contain variola and vaccinia. Variola is the cause of smallpox. Vaccinia is cowpox. And if you remember, back in the late 1700s, Edward Jenner was the first one to come out with the true first vaccine. And what he used was scrapings from lesions of cows that had cowpox. And because smallpox and cowpox are so closely genetically related, Jenner actually used the cowpox, quote, vaccine to vaccinate a young boy against smallpox. And in fact, the term vaccine actually comes from the Latin for cow, which is vacca. The herpes viridae family contains the simplex viruses, and this is herpes simplex type 1 and herpes simplex type 2. Herpes simplex 1 causes fever, blisters, and cold sores, primarily in the oral cavity and nasal passages, whereas herpes simplex 2 causes genital herpes. The herpes varicella zoster virus, primarily herpes varicella causes chickenpox, while the herpes zoster causes shingles. And then the human cytomegalovirus virus, which causes cytomegalovirus infections in humans. And there's one other in the herpes viridae family, and that is the Epstein-Barr virus that causes mononucleosis, among some other things. Adenoviridae family, primarily human adenovirus causes respiratory infections with a few GI infections. Papavirinae family contains the papillomavirus and the CJ virus. Hypnoviridae family contains hepatitis B, and all of the hepatitis viruses are actually all in different families. The Picornaviridae family contains poliovirus and the Coxsackie virus, which causes hand, foot, and mouth disease in humans, and the hepatitis A virus. The Caliciviridae family contains norovirus, which causes a severe gastrointestinal disease, sometimes known also as Norwalk virus. Togaviridae family contains the encephalitis viruses, which includes eastern and western encephalitis, equine encephalitis virus, as well as St. Louis encephalitis virus. All three of these are also found in equines, but also infect humans, and all three are vectored by mosquitoes. Togaviridae family also includes the rubella virus, which causes the German measles. The Flaviviridae family contains dengue virus fever, which is an up-and-comer around the world, West Nile virus, and yellow fever virus. The Coronaviridae family. Oh, have we heard enough about this particular family recently? The coronaviruses, yes, typically cause respiratory disease. Some of them can actually cause enteritis and a GI problem. And of these coronaviridae, of the beta coronaviruses, this includes the SARS and the current coronavirus. The Filaiviridae family contains Ebola and Marburg virus. Orthomyxoviridae family, these are your influenza viruses. Paramyxoviridae family includes parainfluenza virus, the mumps virus, and the measles virus, as well as the respiratory syncytial virus, which is one of the common cold viruses. Now, the Rhabdoviridae family, this name has actually changed recently to Rhabdoviridae, and this is the one that contains rabies virus. Of the Bunyaviridae family, I will just simply mention this one, the Crimean Congo hemorrhagic fever virus. And the reason I mention this is because this is actually a new emerging disease in tropical regions, and it is becoming a larger concern around the world at this moment. The Viridae family, the big one here is rotavirus, which causes GI infections in humans. It's estimated that everyone under the age of 5 in the world has had at least one rotavirus infection, if not more. And in undeveloped parts of the world, rotavirus is the #1 killer of children under the age of 5. The Retroviridae family in the genus of the lentiviruses, this is HIV and causing acquired immunodeficiency. We also have now a Retroviridae virus that is an up-and-comer, and that's human T lymphotrophic virus 1 that is associated with a T-cell leukemia cancer. Arenoviridae family contains the Lassa virus, which is also a fairly emerging disease in tropical regions of the world. So, standard virus classification systems use all of the following except for what? Genetic makeup, the structure, the disease it causes, or the chemical composition. A virus name ending in viridae denotes a viral what? Genus, family. Now, how do animal viruses replicate? Well, there's general phases in the life cycle of animal viruses, and we'll go through these. First of all, we had adsorption, that's not absorption, but adsorption, penetration, uncoating, synthesis of the virus, assembly of the virus, and then release from the host cell. Adsorption is simply where the virus attaches to its host cell by specific binding of its spikes to some sort of cell receptor. So adsorption is the actual binding of the viron to the host cell. Penetration and uncoating can occur at the same time. Penetration, the virus is engulfed into a vesicle and its envelope then will be uncoated. We have to break open that capsid envelope, thereby freeing the genetic code into the cell's cytoplasm. Once in the cell, then depending again upon what type of virus it is, whether it's RNA, DNA, positive sense or negative sense, we then will see synthesis, and this is replication and protein production of viral proteins. Under the control of the viral genes, the cell now is hijacked to synthesize the basic components of new viruses, both the genetic code as well as the capsid proteins and proteins that will make up the spikes. Then once everything is made, the virus spike proteins are inserted in the cell membrane of the cell envelope. The nucleocapsid is formed from the RNA and capsomeres, and then the cell is released from, the virus then will be released from the host cell through either budding or by the host cell actually rupturing. And again, this figure just shows, are you a positive sense RNA, negative sense RNA, double-stranded RNA, or are you a retrovirus in the RNA carrying viruses and how they actually replicate within their host cell? With DNA viruses, if you're a double-stranded DNA, then all we're going to do is simply copy both strands of the DNA to create new copies of the viral genome. We're going to then make messenger RNA from the double-stranded DNA, and that messenger RNA then will make viral proteins. If you're carrying a positive sense DNA genome, this will actually just simply be copied to a double strand, and again, then we will copy the negative sense strand to make the positive DNA strand for viral replication, and we'll make a messenger RNA strand that would be translated into viral proteins. Invasion of the virus begins when the virus encounters a susceptible host and adsorbs specifically to receptor sites on the cell membrane. So adsorb is to attach, like a virus. And so we will find receptors, usually for something else, on the surface of these host cells. And these spikes will actually bind to those receptors and typically then will cause endocytosis to occur. So make sure you know the difference between adsorb and absorb. Host range actually varies. A virus can invade its host cell only through making an exact fit with a specific host molecule on the surface of the host cell. Most viruses are have a restricted host range. For example, hepatitis B only infects liver cells of humans because the human liver cell is the only cell that has the receptor that hepatitis B can adsorb to. Intermediate host range, such as poliovirus, this can infect intestinal and nervous cells of any primate. And broad range hosts, this means that a virus typically will be able to infect cells across many species. And a good example here is rabies virus. Rabies virus can infect the muscle cells and neurons of any warm-blooded mammal. Now, once the virus is adsorbed to the receptors, this usually will trigger then endocytosis. The entire virus will be engulfed by the host cell and enclosed in a vacuole or vesicle. Enzymes in the vacuole then dissolve the envelope in the capsule, if they have an envelope, and release the viral genome into the cytoplasm. So here we've adsorbed. We then trigger endocytosis and uncoating once we are in the host cell. The viral nucleic acid then takes control over the host's synthetic and metabolic machinery. It basically hijacks the host cell's own genome. Mechanisms vary depending on whether the virus is a DNA or RNA carrying virus, and again, whether they're double-stranded, single-stranded, positive sense, negative sense. RNA viruses replicate in the cytoplasm, whereas DNA viruses must travel to the nucleus and replicate there. So again, some same pictures. The RNA virus here, depending again upon whether it's a positive or negative sense strand, will actually have to be modified within the cell. So if you're a positive RNA carrying virus, we're simply going to use that to make a peptide, whereas we also can copy it to a negative sense strand and make its opposite copy to positive sense. That positive sense strand and the viral proteins will be assembled into the new virus. If you're a negative sense strand RNA carrying virus, this can actually just be copied to positive sense so that we can use that for translation to the proteins, and it can be used to make the negative RNA strand that we can then assemble into the new viron. If you're a double-stranded RNA, we're going to simply then use the positive strand to do our proteins and then copy that to make the double-stranded RNA for the new viron. If you're a retrovirus carrying the reverse transcriptase virus, we'll take that positive strand RNA, make a negative DNA strand, and then come back and make the positive sense DNA strand. So that now we have a double-stranded complementary DNA strand, and then we can use that just like we see in any transcription translation process and make our messenger RNA strands. And this is a nucleus of a eukaryotic cell containing hundreds of adenovirus virons. So all of these little spots are actually the virus that's being made. Now, release of the mature viron particles usually occurs in animal cells either by viral budding or by exocytosis. The nucleocapsid binds to the membrane. A small pouch is formed, and it pinches off this pouch, releasing the virus with some of the cell membrane, or again, nuclear membrane, and this will become the envelope of the cell, or envelope of the virus. The viruses then are shed gradually without the destruction of the host cell until it reaches a point that the virus can no longer support it. So we simply see this process of exocytosis. Cytopathic effects include virus-induced changes to the cell that alters its microscopic appearance. The host cells can become distorted, undergo major changes in their shape or size, or develop intracellular damage to the point that they eventually die. In some viruses, we form inclusion bodies, which are compacted masses of virus or damaged cell organelles in either the nuclear plasm or the cytoplasm. Or some viruses actually form what we call syncytia, and this is where the host cells actually fuse together to form large single cells containing multiple nuclei. So here's a normal host cell right here, and then this is one of these syncytia cells. So all of these cells have fused together. We think syncytia may actually form in trying to fight off the virus. In this picture, we actually see inclusion bodies, and these are just simply clumps of the virus, or again, damaged organelles within the cells. This table just shows some of the cytopathic changes with some of the viruses in animal cells. Now, a carrier relationship can develop with some cells, with viruses. The cell can harbor the virus, but is not immediately destroyed by the virus. A provirus is simply where the viral DNA becomes incorporated into the DNA of the host cell, where it can sit silently, sometimes for years. This then sometimes results in a chronic latent state where we get periodic activation after a period of viral inactivity. I have herpes simplex 1, and occasionally I will get an outbreak of fever blisters on my lip. This is because there are neurons that are chronically infected with the viral genome of the herpes virus, and this is simply then being harbored in these cells. But when I don't have active lesions, it is not an actively recurring virus at that point. Now, what about viruses and cancer? Well, oncogenic viruses. Oncogenesis means formation of cancer. It's estimated currently that up to 20% of all human cancers are caused directly by viruses. One of the things that we know about cancer is that cancer cells become cancer cells due to a change of the genetics within that host cell. So if viruses enter the host cell with their genome, does that not change the host cell's genetic makeup? And the answer is actually yes. So virus-carrying genes that can directly cause cancer. The virus can produce proteins that induce the loss of growth regulation in the host cell, and this can cause the host cell then to transform into a cancer cell. So, for example, especially some of the retroviruses, because the complementary DNA that is made from the viral RNA is actually spliced into the host's own genome, this actually can then become a viral oncogene in the provirus state. Now, when we're reading this viral code, we're producing viral proteins. But because it has actually changed the viral or changed the code of the host cell, this may actually lead to uncontrolled cellular growth and the formation of cancer. Other retroviruses, the viral gene affects expression of the host's oncogenes. Yes, we have cancer-causing genes already in our genome. Most of the time, these genes are inactive and not being read. But because of the presence of the viral genome, this can actually turn these human oncogenes on, leading to uncontrolled cellular growth. DNA tumor viruses, those that can carry viral DNA, or some that carry viral DNA, can actually simply then produce viral proteins that themselves actually are oncogenic in nature. Now, viruses that infect bacteria, these are the bacteriophages, and these were just first discovered in 1915. They parasitize every known bacterial species, even though most bacteriophages are very species specific for their bacteria. Often make the bacteria they infect more pathogenic to humans, however. The T-even bacteria, such as the T4 bacteriophage, infects E. coli. And this is T2, T4. And again, these are kind of these weird shaped viruses. These are the most widely studied bacteriophage because we can certainly make lots of E. coli in the laboratory in growing it, and so we can study this very easily. They go through similar stages as animal viruses. They actually will inject the viral genome into the bacterial cell. And in this photomicrograph, you can actually see these green lines right here. And this is actually the DNA that's being injected from the nucleo capsule into this bacterial cell. And this is, again, just another photomicrograph of these being injected into this now dying bacterial cell. The bacteriophage actually can go through two different phases of its life cycle. The lytic phase, or lytic cycle, is the life cycle of the bacteriophage that ends in the complete lysis or destruction of the bacterial cell. The lysogenic cycle, however, the bacteriophage's genome becomes incorporated into the bacterial host cell's own DNA, and the DNA then will actually be copied to subsequent generations of that cell. So here's what it looks like. So in the lytic stage, we'll see that the bacteriophage infects the E. coli. And we make then, or the bacterial DNA and the viral DNA are incorporated. Typically what will happen is that the bacterial chromosome will be broken up by the presence of the viral DNA. This then will cause the protein mechanisms to make viral proteins only. We assemble the new virons, the virons maturate, and then we'll actually lyse the cell. However, sometimes the viral DNA will actually be just simply spliced into the E. coli's own chromosome. And what that means is that as the bacteria then undergo cellular division, this piece of the DNA, the viral genome, will actually be copied then to the next generation. At some point, however, from the lysogenic stage, it then will then revert back and go back into a lytic stage and actually cause then release of huge numbers of the virus. Temperate phages have the ability to undergo adsorption and penetration, but do not immediately undergo replication or release. They basically form, again, this prophage, which is the inactive state of the viral genome, in which the phage DNA is inserted into the host's own chromosome during lysogeny. Now, induction has to occur then for the activation of the prophase in a lysogenic cell to then enter the lytic cycle and to reproduce the virus. And we don't always know what actually causes this transformation from the lysogenic phase into a lytic cycle again. The danger of lysogeny in human diseases is that occasionally phage genes from the viruses found in the bacterial chromosome cause the production of toxins or enzymes that actually cause pathology then in the human, making the bacteria more pathogenic. Lysogenic conversion, with the acquisition of the new trait from one of these temperate bacteriophages, is actually been linked to responsibility for the production of the diphtheria toxin, cholera toxin, and actually botulism toxin. It's estimated that bacterial genomes are probably about 20 to 25% actually viral genomes that have become incorporated in the lysogenic conversion and actually now become part of the bacterial's own genome. So this table just compares the bacteriophage and the animal virus multiplication. So can you list and describe the main steps of animal virus replication? Can you tell the difference between DNA virus replication and RNA virus replication? And true or false, viruses that infect animal cells can never infect human cells. And describe the difference between lytic and lysogenic phase of viral replication. Now, viruses cannot be cultured like bacteria within the laboratory. We can only replicate viruses in living host cells. So in vivo, literally meaning in life methods, viral cultivation has to be done in either laboratory animals or embryonic tissues of some sort. And usually we use chick embryos. In vitro, which means not in the living organism, the viral cultivation is done in cell or tissue cultures. The viruses have to get inside living host cells, but these cells or tissues are actually grown in artificial culture. Isolation and identification of viruses in clinical specimens is the primary purpose of viral cultivation. But the other big thing is that we can prepare viruses for vaccines. We can do detailed research on the viral structure, their multiplication cycles, their viral genetics, and the effects that they can have on host cells. Specifically bred strains of animals are typically used for animal cultivation of viruses so that we know the genome of the animals that we are using. And typically, historically, we've used the white mice, we've used rats, hamsters, and even guinea pigs. Some rabbits have been bred for viral application as well. And in fact, Louis Pasteur actually used rabbits to produce that first rabies vaccine. Injection sites for viral exposure in laboratory animals can be in the brain, the blood, muscle tissue, into a body cavity, or even in the skin itself or their foot pads. Now, using chick embryos, first of all, the embryonic development occurs in a protective shell, and that's a big advantage so that we don't get any contamination. The bird embryo is an intact and self-supporting unit with its own sterile environment and nourishment, and it can furnish several embryonic tissues that support viral multiplication. Cell culture or tissue cultures, the cells and tissues are grown in vitro versus a living cultivation system such as the bird embryo or laboratory animal. This is a simple and effective way to grow populations of isolated animals in culture dishes. Most viruses are now propagated through cell cultures, and much of a virologist's work involves developing and maintaining these cell and tissue cultures. So here's a microscopic view of an undisturbed layer of animal cells, and this looks like probably an epithelial cell culture of some sort. Whereas now what we'll see is these are now infected animal cells within this culture. So the open areas are the spaces where the cells have been disrupted by the virus infection itself. Primary cell cultures are freshly isolated animal tissues that are placed in a appropriate growth media, and the cells will undergo mitotic division, producing a single layer or monolayer of the tissue cells. They retain the characteristics of the original tissues from which they were derived. Continuous cell cultures have altered chromosome numbers. They typically can grow very rapidly, and they can show changes in the morphology of the cells. They can be continuously subcultured if they are given fresh nutrient media. Detecting viral growth in cultures, usually we're going to see degeneration and lysis of the infected cells, showing again those empty spaces like we just saw. In some cases, we'll form plaques, which are clear, well-defined patches in the cell sheet. And these develop when the viruses are released from their infected cells and radiate out to the surrounding cells to infect them. Infections spread typically gradually and symmetrically from the original point of the site of infection. So here's our next concept check. Can you describe the primary purpose of viral cultivation? The influenza virus has traditionally been cultured in what? And this is a problem with our influenza vaccine, is that we cannot give the vaccine to certain people that have an allergy to this particular substance in which the influenza virus was cultured. And can you describe the differences between primary and continuous cell lines? And true or false, the term plaques is only used to describe the effects of bacteriophage on cell cultures. And now we'll talk about the other non-cellular infectious agents. This includes the spongiform encephalopathies. This is a prion diseases, and this has been implicated in chronic persistent disease in both humans and animals. Brain tissue removed from affected animals or humans resemble a sponge, hence the name spongiform encephalopathies. Prions are a common feature of the spongiform encephalopathies. It's a distinct protein fibrils that are deposited in the brain tissue of affected animals. Creutzfeldt-Jakob disease, or CJD, is the form that affects humans. It affects the central nervous system in humans and causes a gradual degeneration of the central nervous system and eventually death. It's transmissible by an unknown mechanism, but there have been models that have been proposed that it could be from actually eating infected brain tissue either of other humans or other animals. Several animals are victims of similar diseases, and this includes scrapie in sheep, mink, and elk, the bovine spongiform encephalopathy or mad cow disease in cattle. And what we actually will see is that there are simply holes that will appear due to the death of the neurons that are being affected in these prion diseases. And this is actually a classic characteristic of CJD disease. This is what bovine spongiform encephalopathy looks like. The cows simply become uncoordinated and eventually go completely down. That is why in the United States, no downer cow can actually go into a slaughter plant. Now, there's lots of reasons why a cow will go down and not be able to get back up, but because of the danger of bovine spongiform encephalopathy that could get into humans, this is one of the reasons why no downer cow will go into slaughter plants. And this is actually what scrapie looks like in sheep. And you'll notice that one of the reasons it's called scrapie is because they literally lose all of their wool or hair in the mink or the elk. Prion infections, the exact mode of infection is actually unknown. We know, however, that prions are proteins that actually act as a virus. So it's a protein composition of the prion that has revolutionary ideas of what can constitute an infective agent. So I literally think of prions as a proteinaceous structure that acts similar to a virus. Questions about how these prions replicate, given that they have no nucleic acid, is still up for debate. Satellite viruses are dependent on other viruses for replication, and one of these is the adeno-associated virus that has to actually replicate inside a living cell that has an adenovirus. Originally thought that this virus could only replicate in cells infected with the adenovirus, but recent studies have actually shown that to infect cells infected with other viruses, or that they've had their DNA disrupted through some other means. Viroids are virus-like agents that parasitize plants only. These are actually about 1/10 the size of an average virus, and they are composed only of a naked strand of RNA. However, they lack a capsid or any other type of coating. So viroids are simply RNAs that infect plants that cause virus-like diseases. Significant pathogens in economic important plants such as tomatoes, potatoes, cucumbers, citrus trees, and chrysanthemums. So, what are prions composed of? DNA, RNA, proteins, or polysaccharides? Satellite virus requires what in order to multiply? And what is the substance that infects only plants that is made up of naked RNA? Now, it is impossible to measure the number of viral infections that are seen worldwide. Most common cause of acute infections that do not result in hospitalization are going to be viral in nature. And this would include things like colds, chickenpox, influenzas, herpes, warts, etc. Viral agents that only occur in certain regions are like dengue fever, Rift Valley fever, yellow fever, and oftentimes this is because the virus is vectored by a particular vector found only in those certain regions. Some viruses have very high mortality rates. For example, rabies is 100% fatal with no treatment. And yes, human rabies does still occur in the United States. Ebola, overall, of all the strains of Ebola, it has about a 50% fatality rate overall. The original Ebola, however, was almost 90% fatal. It wasn't until it had actually mutated that some of the new strains had become a little less fatal with about a 60% mortality rate. And other viral infections can lead to long-term disabilities, such as polio. While polio has pretty well been eradicated throughout the world, there are still a number of places with very high populations that still see polio on a daily basis and is considered endemic in those areas. And unfortunately, these children that have had polio, these effects are not going to go away. In fact, most of the time, they actually worsen as the person ages. And also neonatal rubella. If the mother actually gets rubella during pregnancy, this can lead to some long-term effects in the fetus. Viruses mutate at an extremely rapid rate. It is difficult to design therapies, therefore, against all strains of viruses. Scientists focus on developing vaccines against viruses since so few antiviral drugs are available and antibiotics are completely ineffective against viral diseases. Antibiotics only work against bacterial diseases and not viral diseases. One of the areas of interest is something called interferon, and this is a naturally occurring cell product that is produced by virally infected cells. Interferon interferes with the development and reproduction of viruses within subsequent cells. And so these have actually been used with some success in preventing and treating viral infections, especially in humans. So, viral infections are more common than bacterial infections. True or false? Viral mutation rates are low and viruses are easy to track within populations. True or false? Doctors and scientists most often rely on which of the following to treat or prevent viral infections?